Picture this. A group of elephants is moving through the African savanna in the particular purposeful way that elephants move when they have a destination in mind. Not the relaxed grazing drift of a group with no agenda. The focused, coordinated locomotion of animals that know where they are going and are going there with intent. The destination, it turns out, is a grove of marula trees. Trees whose fallen fruit, fermenting on the ground in the heat, has reached a sugar to alcohol conversion stage that the elephants in this region have been tracking, visiting, and returning to with a regularity that goes well beyond the behavior of animals simply seeking nutrition. The elephants arrive, they eat the fermented fruit. A lot of it. More than caloric need would require. Then, over the following hour or so, something shifts in the group's behavior that any observer who has spent time around drunk humans would recognize immediately. A loosening of the usual social precision, a slight but perceptible change in coordination, a quality of increased vocalizing and social contact that looks, to any honest observer, less like a group of animals that happened to consume some fruit, and more like a group of animals that consumed some fruit on purpose and are now experiencing the expected results on purpose. The elephants are not confused. The elephants are not accidentally eating fermented fruit because fermented fruit is what was available and they had no preference. The elephants sought this out, they remembered where the marula trees were, they traveled to reach them at the right time, they consumed quantities that go beyond what nutrition requires. And they are doing it again next season and the season after that with the kind of behavioral consistency that, in any other context, would be described as a habit. Welcome to one of the most entertaining and most scientifically revealing topics in the entire field of animal behavior. The animals that actively, repeatedly, and in some cases with considerable ingenuity seek out psychoactive substances. Because it turns out that humanity's complicated relationship with substances that alter the state of the brain is not a uniquely human complication. It is a behavior so widespread across the animal kingdom, so consistent across species that have no evolutionary relationship to each other, and so persistent in populations that have been observed across extended time periods, that it raises questions about the neurobiology of reward, the evolutionary origins of substance-seeking behavior, and what exactly it means that the brain systems underlying intoxication are old enough to be shared across species separated by hundreds of millions of years of evolution. This is the full, properly examined story of which animals cannot stop getting high, why they do it, what it reveals about the neurobiology they share with us, and what the ancient humans who observed these behaviors across thousands of years of close contact with the natural world made of the animals around them that seemed to be doing the same things they were doing with plants and fermented substances, with considerably less social complication. Start with the neurological foundation, because understanding why animal intoxication-seeking behavior is so widespread requires understanding what psychoactive substances are actually doing in animal brains and why those brains respond to them the way they do. Psychoactive substances, whether alcohol, opiates, cannabinoids, stimulants, or hallucinogens, produce their effects by interacting with receptor systems in the brain that evolved for entirely different purposes. The opioid receptors that respond to opiates evolved to respond to the brain's own endogenous opioid molecules, endorphins, and related compounds produced internally during physical exertion, pain, and social bonding. The endocannabinoid receptors that respond to cannabis compounds evolved to respond to internally produced cannabinoid molecules that play roles in appetite regulation, pain modulation, memory, and stress response. The reason external psychoactive substances produce such powerful effects is that they interact with these receptor systems at intensities, durations, or specificities that the endogenous molecules do not achieve. They essentially hijack systems that evolved for internal regulatory purposes and stimulate them at levels or in patterns that the systems were not calibrated to handle. The result is altered perception, altered mood, altered cognition, and in many cases altered motivation, effects that are experienced by any organism with the relevant receptor systems, regardless of whether that organism is a human attending a party or an elephant eating fermented fruit off the ground. The critical implication of this for the animal intoxication question is that these receptor systems are ancient. Opioid receptor systems appear in organisms as evolutionarily distant from humans as fish. Endocannabinoid systems appear in invertebrates. The neurochemical infrastructure for responding to psychoactive compounds is not a human specialization, or even a mammalian specialization. It is distributed across the animal kingdom in ways that reflect its ancient evolutionary origins in basic neurological regulatory functions. Any animal with the relevant receptor systems is, in principle, capable of experiencing the effects of substances that interact with those systems. And any animal with a brain capable of associating the consumption of specific substances with the subsequent altered state, which includes most animals capable of learning from experience, is capable of developing the behavioral patterns of returning to those substances to repeat the experience. Now, move through the specific animals where this behavior has been documented most extensively and most interestingly, because the range is extraordinary and the specific details of how different species pursue intoxication are both entertaining and scientifically revealing. Reindeer and psychedelic mushrooms represent one of the most culturally resonant and most behaviorally consistent examples of animal intoxication seeking in the documented record. Amanita muscaria, the fly agaric mushroom, the red-capped, white-spotted mushroom of fairy tale illustration, contains psychoactive compounds including muscimol and ibotenic acid that produce hallucinogenic and sedating effects in mammals. In the boreal forests and tundra environments where reindeer live, fly agaric mushrooms grow in association with birch and pine trees and are encountered regularly by reindeer during their foraging movements. Reindeer consume fly agaric mushrooms with a behavioral enthusiasm that goes well beyond what nutritional content would justify. They actively seek the mushrooms out. They compete with other reindeer for access to them. They consume them preferentially over other available forage. After consumption, affected reindeer show behavioral changes: unusual vocalizations, altered gait, apparent perceptual disturbance, social behavior that differs from baseline, that are consistent with psychoactive intoxication, and that observers across different cultural and research contexts have described in ways that converge on the same basic interpretation. The reindeer are high, they sought to become high, and they will seek it out again. The cultural dimension of reindeer-mushroom interaction in the circumpolar regions adds a layer that connects this animal behavior to ancient human practice in a fascinatingly direct way. Indigenous Siberian cultures, with ancient roots in these environments, documented the relationship between reindeer and fly agaric over centuries of close observation of the animals they lived alongside, herded, and depended on. In these cultural contexts, fly agaric also had human ceremonial and shamanic use, creating a situation where ancient humans and reindeer were, in some respects, utilizing the same psychoactive resource in the same landscape, a convergence that did not go unobserved by the people involved. The urine recycling behavior associated with fly agaric consumption in both reindeer and humans in these traditions is one of the more unusual specific details in this story, and is documented across multiple independent sources in ways that make it difficult to dismiss as folklore. The active psychoactive compound muscimol is excreted relatively intact in urine after consumption. In environments where fly agaric mushrooms were scarce or seasonal, consuming the urine of an individual who had recently eaten the mushrooms, whether reindeer or human, provided access to the psychoactive compound after the original mushroom was no longer available. Reindeer consuming their own urine after eating fly agaric, and the documentation of similar practices in human shamanic contexts in the same cultural area, represents one of the more specific behavioral overlaps between animal and ancient human intoxication-seeking behavior in any documented tradition. Jaguars and the ayahuasca vine represent another well-documented and culturally significant intersection of animal and human psychoactive plant use. Banisteriopsis caapi, the vine that is the primary ingredient in the ayahuasca preparation used across Amazonian indigenous traditions, is consumed by jaguars in the wild with a behavioral pattern that observers have described as deliberate and repeated. Jaguars chew the bark and leaves of the vine, then show altered behavioral states including dilated pupils, apparent visual tracking of objects or movements that are not perceptible to human observers, and a quality of altered awareness that experienced indigenous observers in the Amazon, people whose knowledge of both jaguar behavior and the effects of ayahuasca preparation on humans extends across generations. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Interpreted as the jaguar intentionally seeking the same altered state that human ceremonial use of the preparation produces. The jaguar's status in Amazonian indigenous cosmology is directly connected to this observed behavior. The jaguar is the primary shamanic animal across numerous Amazonian traditions, the creature most associated with the visionary and transformative dimensions of ceremonial plant use, and the specific association between jaguars and ayahuasca plants. In these traditions reflects generations of close observation of jaguar behavior in the habitat they share. Ancient humans in these environments did not arrive at the use of these plants in isolation from the natural world around them. They were observing what the animals in their environment were doing with the plants available, and the jaguar's relationship with Banisteriopsis caapi appears to have been part of that observational foundation. Cats and catnip represent the most domestically familiar version of this phenomenon, and the one that most people have observed directly without necessarily thinking of it in these terms. Nipetolactone, the compound in catnip that produces the characteristic behavioral response in susceptible cats, interacts with feline olfactory receptors in ways that trigger a behavioral cascade of rolling, rubbing, vocalizing, and apparent euphoria that typically lasts several minutes before a refractory period of apparent indifference sets in, after which the cycle can be repeated. The behavioral response is not learned. It is present in kittens at approximately 6 months of age when olfactory development reaches the relevant threshold, and it is consistent enough across susceptible individuals that it clearly reflects a neurological response rather than a conditioned behavior. Approximately 60 to 70% of domestic cats show the response, with the susceptibility appearing to be genetically determined. Big cats, lions, tigers, leopards show similar responses to catnip and related plants, suggesting the underlying neurological sensitivity is distributed across the felid family rather than being a domestic cat specialization. The specific neurological mechanism is still being investigated, but current evidence suggests nipetolactone may interact with opioid systems in the feline brain, potentially explaining the euphoric quality of the behavioral response and the refractory period that follows it. If the opioid interpretation is correct, cats seeking out and rolling in catnip are, in neurochemical terms, engaging in a form of self-administered opioid stimulation, which reframes the image of a cat rubbing its face ecstatically in a pile of dried herbs in ways that are either more sympathetic or more concerning depending on your perspective. Wallabies in Tasmania stumbled into international attention when agricultural reports from poppy-growing regions documented animals entering legal opium poppy fields, consuming sufficient quantities of the plants to become intoxicated, and then moving through the fields in patterns that created the distinctive crop circles that had previously generated more fanciful explanations. Wallabies under the influence of poppy alkaloids show behavioral changes including impaired coordination, apparent disorientation, and the circular movement patterns that produced the crop circle observations. They also return to the fields repeatedly, which is the behavioral detail that moves this from accidental exposure to something that more closely resembles deliberate seeking. The wallaby poppy situation is interesting from a neurobiological standpoint because opium poppy alkaloids, morphine, codeine, and related compounds interact with the opioid receptor systems that are among the most ancient and most broadly distributed reward-related neurochemical systems in vertebrate neurology. A wallaby experiencing opiate effects is using the same receptor systems that human opiate use effects, because those receptor systems evolved long before either wallabies or humans existed as species. The neurochemical basis for opiate reward is old enough to predate the mammalian radiation, meaning the capacity to experience opioid reward is a shared inheritance across an enormous range of vertebrate species. Horses, cattle, and locoweed represent an intoxication-seeking pattern that has significant economic consequences, and that illustrates an important distinction in this discussion. The difference between substances that are appealing to animals because of reward properties and substances that produce apparent compulsive seeking that may reflect addiction-like neurological dynamics rather than simple reward pursuit. Locoweed plants, various species of Astragalus and Oxytropis, contain swainsonine, a compound that inhibits a specific enzyme involved in cellular glycoprotein processing, and that produces neurological effects, including what appears to be intoxication in horses and cattle that consume it. Livestock that access locoweed once often return to it preferentially, consuming more of it in a pattern that has historically been described by ranchers as addictive. The neurological damage from chronic swainsonine exposure is progressive and cumulative, eventually producing permanent cognitive and behavioral impairment even after the animal is removed from access to the plant. The fact that animals continue seeking locoweed despite the negative consequences of chronic exposure raises questions about whether what appears to be reward-seeking behavior in some animal substance-seeking cases might involve neurological dynamics more similar to addiction than to simple recreational intoxication. A distinction that matters both for understanding the behavior and for the welfare implications. Dolphins and pufferfish represent one of the more recently documented and most behaviorally sophisticated examples of deliberate animal intoxication seeking. Pufferfish, when handled or stressed, release tetrodotoxin, a powerful neurotoxin, but also produce compounds that at low doses appear to have narcotic-like effects. Documentary footage showing young dolphins carefully passing a pufferfish between them, handling it gently in a way that causes the fish to release its compounds without triggering the full defensive toxin response, and then showing apparent altered behavioral states, including floating near the surface in what observers described as a trance-like state, generated significant attention precisely because the behavior showed a level of deliberate, coordinated technique that is difficult to interpret as accidental. The deliberateness of the pufferfish handling behavior, the careful, controlled manipulation that appears calibrated to get the desired chemical effect without triggering a lethal dose, implies a level of learned, transmitted technique that is cognitively demanding. Dolphins appear to learn this behavior socially from other dolphins, which means it is a culturally transmitted intoxication-seeking practice, passing between individuals through the same social learning mechanisms that transmit other dolphin behaviors. A drug use practice with a specific technique that is learned and transmitted socially is approaching a complexity of behavior that, in any other species, would be considered remarkable. Moths and various psychoactive plant compounds represent the invertebrate end of the intoxication-seeking spectrum, with research documenting preference behavior in moth species for flowers producing specific alkaloid compounds, with the preference appearing to reflect something beyond simple nutritional or reproductive benefit. The distribution of psychoactive compound-seeking behavior across invertebrates with nervous systems far simpler than vertebrate brains underscores the point about the antiquity of the underlying neurobiological systems. The capacity to find certain neurochemically active compounds rewarding is not a vertebrate specialization, but a feature of nervous systems broadly. Bees and fermented nectar complete the picture at a level that connects to the earliest stages of alcohol-producing chemistry in nature. Bees exposed to fermented nectar, nectar in which naturally occurring yeasts have converted sugars to ethanol, show behavioral changes including impaired navigation, impaired learning performance, and altered social behavior within the hive. Beekeepers have documented bees returning to hives in states of apparent intoxication from fermented nectar sources, and hive behavior in these situations parallels in some ways the social dynamics of human groups managing members in various states of intoxication, including guard bees at the hive entrance showing differential responses to returning bees in impaired states. The social management of intoxicated members within the hive is documented across multiple independent observations and represents one of the more unexpectedly complex behavioral details in this entire topic. For ancient humans observing the natural world around them across the deep time spans before civilization, the behavior of animals with psychoactive plants and fermented substances was not invisible information. Ancient human knowledge of the natural world was intimate, detailed, and accumulated across generations of close observation of the plants and animals they lived alongside. The animals in their environment were, in some respects, running parallel experiments with the same chemical compounds that ancient humans were beginning to explore, demonstrating which plants produced altered states, which fermented substances produced altered behavior, which mushrooms produced unusual perceptual experiences. The question of whether ancient human discovery of psychoactive plants was in some cases informed by observation of animal behavior with those plants is not definitively answerable from current evidence, but is a hypothesis that several researchers have explored and that the behavioral data from multiple cultures supports as plausible. The consistency of the jaguar-ayahuasca connection in Amazonian traditions is one of the more specific examples. But the broader pattern of indigenous knowledge systems that recognize specific animals as associated with specific psychoactive plants in regions where both the animals and the plants are present and the behavioral connection has been observed over generations suggests that animal intoxication seeking was part of the information landscape from which ancient human plant knowledge developed. The evolutionary question underlying all of this: why do animals seek intoxication at all from a natural selection standpoint, is one that does not have a single clean answer. Several hypotheses have been proposed, and none is universally accepted. The pharmacological byproduct hypothesis suggests that animals seek psychoactive substances for other reasons, nutritional, medicinal, or as a side effect of seeking something else, and that the intoxication is a byproduct rather than a sought outcome. The adaptive intoxication hypothesis suggests that in some cases the altered states produced by certain compounds have adaptive value, reducing anxiety, facilitating certain types of perceptual processing, or providing other functional benefits that outweigh the costs of impaired function during the intoxicated state. The evolutionary trap hypothesis suggests that psychoactive compounds hijack reward systems in ways that were not adaptively relevant in ancestral environments, and that substance-seeking behavior in animals reflects a mismatch between ancient reward circuitry and the specific chemical landscape of their current environment. The honest answer is probably that different substances in different species in different contexts fall into different categories. That some animal substance seeking is closer to the pharmacological byproduct explanation, while some looks much more like deliberate reward seeking, and that the full range of behavior documented across the animal kingdom reflects the full range of ways that the intersection of ancient reward circuitry and available psychoactive compounds can play out. What is not in question is the breadth of the phenomenon. The capacity to find psychoactive substances rewarding is distributed across the animal kingdom in ways that reflect the antiquity and broad distribution of the neurological systems those substances interact with. From reindeer in the boreal forest to dolphins in the Pacific, to wallabies in Tasmanian poppy fields, to cats rubbing their faces in herbs in modern living rooms. The behavior of seeking substances that alter neurological state is not a human invention and not a human problem. It is an expression of neurobiology that our species shares with an extraordinary range of other animals, expressed across an extraordinary range of behavioral contexts, and documented across the full span of human observation of the natural world. Ancient humans watching the animals around them eating the same plants, seeking the same fermented fruits, and exhibiting the altered states that resulted, they were not watching alien behavior. They were watching a version of something their own nervous systems were already telling them to find interesting.